Thank you, Sylvia, for the introduction, and thanks very much, everyone, for taking the time out of your busy schedules to have a listen to what the solutions we have in the NGS arena. Basically, we will be covering the following topics today, and we will be focusing obviously on NGS and target enrichment, and then the solutions that IDT has for this application. So first of all, I'm sure you're all familiar with IDT. Who are we? So we are the largest supplier of oligos worldwide, and we have grown into a global company in the last 30 years. So we have our headquarters in Coralville in the United States, and we have expanded in such a way globally that the sun never sets on IDT. As you always, doesn't matter where you are in the world, you will always have a facility where the sun is shining. So I am based in Leuven. That's a production facility which would be the closest to where you currently situated. So let's first delve into sequencing, and it's a very exciting field to be in. And if you cast your minds back to the 1980s, sequencing started with these gel-based systems with manual slab gels that soon moved into automated slab gels. So this was quite a labor-intensive way of sequencing. The sequencing field evolved into capillary sequencing systems in the 1995s, where ABI capillary sequences sort of flooded the market. And the era of Sanger sequencing started. In the 2005 to 2010s, Roche brought out the 454 sequencer, which essentially used micro-L pyro sequencing and long reads. And after that, the era of Illumina arrived when they acquired a company called Solexa, which had short read sequencing technology. And we then were able to massively parallel sequence and generate a lot of data in a run. So at the moment we are entering single molecule sequencing, and the cost of sequencing is really being reduced dramatically as time goes by, and we can now sequence a human genome for less than $1,000 US, which is quite exciting. So here you can see a comparison in the 2005 compared to today. During the Human Genome Project, there were these large sequencing facilities that needed a lot of capillary sequences, colony picking robots, a lot of manpower to generate the sequences, and it took really a long time to be able to sequence just a short piece of DNA. And now at present, one of these sequencing facilities can be replaced by a single flow cell, and here you can see someone holding an Illumina flow cell. Essentially, sequencing is done massively parallel on this glass surface, and we've entered the era of big data. So generating sequencing data currently is no worry at all. It's how you're going to analyze it. And if you have a enrichment step in your workflow where you are able to enrich for specific target sequences, you are able to generate more useful data that will allow you to confidently call more mutations. And I will talk later on about the solutions that IDT has just to allow you to do the target enrichment. So just to make sure that we're all on the same page, I'm just going to put up the basic NGS workflow, and this is the major steps that you would find in an NGS workflow. There will be some changes based on different applications, but just for argument's sake, you would normally start with DNA as input. So DNA obviously is a long molecule, and it needs to be broken down into smaller pieces to be able to manage it and to be able to generate shorter reads massively parallel. Now DNA needs to be fragmented, and there are several ways of doing that, and that is either via an enzymatic or mechanical step. Once the DNA is broken into smaller pieces, you have to make that adaptable to be manipulated on the sequencer, and this is done by incorporation of adapters. Once you have your library containing adapters and you want to do an enrichment step, a target capture or an PCR-based amplification step would follow. Here we're just focusing on hybridization. And this would then be followed by adding your library or your enriched library on a sequencer. And depending on the sequencer that you use, you would go through, for instance, an emulsion PCR or a bridge amplification step, followed by sequencing, and then primary, secondary, and tertiary analysis. So let's just look at the different steps during the library prep step in the sequencing workflow. And this is, as I mentioned, the first step would be to fragment your DNA. So it will be broken up into smaller pieces to make it more manageable. And there are two ways of doing this, and this is via mechanical or acoustic shearing, where you can use a Cavaris instrument to randomly fragment your DNA, or via enzymatic shearing step. And there are two ways of doing that. And this is either via fragmentase that will randomly fragment your DNA, or a transposase. Now, a transposase will basically do two things. It will fragment your DNA and also incorporate smaller adapter sequences that can be recognized by primers containing indexes and the rest of your adapter sequences. In a subsequent PCR step, this can be incorporated, and you will be left with your sequence-ready library. And the transposase system is an example, or one example would be the Illumina Nextera kits. So once you've fragmented your DNA, as I mentioned, you have to add your adapters, and this can be done in two ways. Here we are ligating our adapters to our fragments, which will then allow you to have a sequence-ready library to be able to manipulate it on the sequencing instrument. So once you've added your sequence-ready library to your sequencer, you will go through an amplification step. So you have to amplify the signal. So here I'm explaining the Illumina workflow. So here it is sequencing by synthesis, and this is what happens on a flow cell. So here you can see on the left-hand side, you can see one fragment that has already been hybridized to the flow cell with a capture oligo and then extended. So now it's covalently bounded. So here you have one single fragment. If you incorporate the base that's fluorescently labeled onto the flow cell now, the signal will be too weak to be able to distinguish and detect the signal. So you have to amplify the signal, and the way it is done is by cluster generation or bridge amplification. So basically, the fragment will flop over and find its mate, and you will then basically do an extension, and this will be done several cycles to generate a cluster. So you're making more of a specific single fragment. The way you can also look at this is by comparing it to a petri dish where you have bacterial colonies that grow on a flow cell. When one bacterial cell finds a place that it can establish itself and start to multiply, it will make clones of itself. And this is essentially what cluster generation it does. It makes more clones of a single fragment. And you normally will strip off the antisense strand and only for the first round only sequence the sense strand to be able to make sure that you have the same base incorporated for the entire cluster. So once you have your clusters generated, you're sequencing the sense strand, you will add your sequencing primer, and then you will also add in several cycles one base at a time, which is fluorescently labeled. So you have all bases, actually all four bases are added simultaneously, and they're fluorescently labeled, each with a different dye and blocked for extension. So in each cluster, on each strand, only a single base can be incorporated, and you will then detect it via a fluorescent signal. And for that specific cluster, for an XY coordinate on a glass slide, you will take a picture and build up your sequence as you go along. So you can see one cluster, the T is incorporated, you see a blue fluorescent signal. As you go along, you're incorporating a G, C, T. So based on the pictures, for that specific XY coordinate, you build up your sequence. And this is done massively parallel for each cluster. So to be able to do this, you need a sequencing platform, and as I mentioned, Illumina has quite a large portfolio of sequences to cater for all your needs. So depending on your throughput and your application, you have several platforms to choose from. So we start from the recently launched MiniSeq, that's the lowest throughput sequencer, to the High-seq 2500, which produces about 1 terabase per run. There's also a system that they have available called the High-seq X10 that is used for population sequencing, so very high output. So depending on your needs, you choose a specific sequencer, and for target enrichment, you obviously will not enrich an exome and sequence it on a MiSeq or a mini-seq. You will basically go over to the exome for an exome on a NextSeq or a High-seq. So Thermo Fisher also have the ion torrent sequences available. Also you can see the portfolio. The instruments that are used most commonly out in the field would be the Proton and the PGM. And these have a bit lower output capability than the Illumina sequencers, but these are also conducible for targeted sequencing when you have smaller panels. So once you've done your library prep and you want to enrich for specific targets, you can go through a target enrichment step. Now, one thing that I want to mention with regards to target enrichment is that it is not 100% effective. So it's a very dirty process. Ideally, in a perfect world, target enrichment would be, if you're looking for 1,000 targets, you'd cast out 1,000 fishing rods with the same bait targeting a species, and when you pull it in, you would have caught your 1,000 specific target species. Unfortunately, it does not work that way. It's basically casting a net out at a specific depth in the ocean and targeting a species and hoping for the best. So you will always have some non-specific sequences that come and is enriched simultaneously. So when you have a very small panel, we have seen that your on-target percentages is lower than when you have a larger panel. The on-target percentages would be a bit higher. So for an exome panel, you can expect very high on-target percentages. Why would you do target enrichment? Obviously, it's self-explanatory. When you're enriching for a smaller target region, you have the capability of multiplexing at higher levels, which will allow you to produce more significant usable data at a higher read depth to allow you to also more confidently call mutations and detect variants. Two ways currently of performing target enrichment is via PCR-based amplification. And this is a product that we are working on, and we will have an amplicon-based enrichment system available for you soon, so watch this space. One of the focuses of this NGS presentation is, or webinar is, target enrichment based on hybridization capture using our lockdown probes, and I'll talk you through that more in subsequent slides. So hybridization capture is using specific baits that will hybridize to specific target sequences and pull those out from a specific genomic background. So this is an example of target enrichment via amplification. The basic steps would be that you would go through a locus-specific PCR that contains flaps with adapter sequences. You would do the first initial PCR, and in a subsequent PCR, you would use primers that will bind to the adapter sequences and will incorporate the indexes and the rest of the adapter to make the library ready for sequencing. Conversely to that, we have in-solution hybridization. So this is target enrichment using hybridization probes. And I do need to mention at this point that IDT allows you with our capture protocol to use any library prep kit of your choice. So you have flexibility to use a library prep kit that you've already optimized for your specific sample type. All you need to know is that you need 500 nanograms of pooled indexed library. So this means if you have one target that you want to enrich for, one sample, you would use 500 nanograms of that library. If you have 10 samples that you want to multiplex together and pull together, you would use 50 nanograms of each to make up the 500 nanograms. You also need to know that you have to have the full-length adapter, so depending on the platform that you're using, we need to know which adapters you're using, what is your indexing scheme, so that we can recommend the corresponding blocking oligos. The blocking oligos that IDT supplies are universal, and they contain proprietary modifications. And what these do is actually bind to your adapter sequences so that it inhibits hybridization of adapter sequences during capture. So the adapter sequences are obviously the same, and you have several fragments, some containing target sequences, some that don't. Or you obviously don't want adapters of a target sequence fragment to hybridize to a region that has a non-target sequence, because that will basically, when you do a pull down, enrich for non-target sequences. So you'll have a daisy chaining of non-specific sequences that get pulled through during your capture. So the blocking oligos can improve your on-target percentages by inhibiting the binding of adapters to each other. Then you will have a panel. So a panel you would buy from IDT. We would do the design, and the panel will consist of xGen lockdown probes. So this will be essentially 5' biotinylated 120-mer DNA oligos. So you will hybridize that to your library and do a normal strip biotin magnetic pull down, and you end up with your enrich library. So a very quick protocol, and this is actually the IDT protocol that we show here, and you can complete the protocol in a day's time. So to summarize the solutions that we would have, and this is a busy slide, so IDT solution can be broken up into two parts. So first of all, library preparation, and secondly, target enrichment. So as I mentioned, for library prep, we do not have a kit available, but you're open to using any kit of choice. There are some exclusions that we can talk and some workarounds that we can talk about at a later stage. But obviously, as IDT is an oligo supplier, we can cater for all your oligo needs. That is in your library prep workflow, and also all your adapter needs. So if you want to incorporate your adapters via PCR, or you want to incorporate your adapters via ligation, we have solutions available for you. You just send us the sequences, and we will synthesize and include the necessary modifications. And I'll talk more about the modifications needed a bit later. True grade processing would also be something that you would be interested in if you are looking at clinical diagnostics and rare variant detection. True grade processing is adapter synthesis technology or procedure where we synthesize adapters to prevent index crosstalk, and I'll talk a bit about that a bit later. So our target enrichment using our capture probes, we have several options available, and here you can break these down into custom or pre-designed. So custom would be for all species other than human, and all content that it would not be present in our pre-design. So pre-design would be for the human species, and here we have on the pre-design two solutions available, and that would be either stocked panels, including an exome panel, or gene capture pools. And gene capture pools would be targeting specific coding regions or coding regions of almost all human genes that you can select from a list. We also have, as I mentioned, our universal blocking oligos. We have our own buffer and hybridization reagents. And if you're interested in NGS controls, please feel free to contact my colleagues Sylvia and Stella. Our G-blocks would be double-stranded, sequence-verified pieces of DNA that are delivered at specific concentrations, and you can use these to sort of barcode your samples if you're doing high level of multiplexing, just as an added step of insurance. Okay, so let's delve a bit deeper into the solutions for library prep. And as I mentioned, we can cater for all your adapter needs. I obviously don't need to remind you what an adapter looks like, but just for the sake of being complete, I would just mention the three parts that adapter would normally include, and that would be a solid support binding in red. This would normally bind to a flowcell or a chip. This allows manipulation of your fragment on the solid matrix or solid support. You obviously will have a barcode if you want to multiplex samples together. This basically marks specific reads to specific samples, and then your sequencing primer, which will allow this and facilitate sequencing of your library fragment. Now let's say you want to incorporate your adapters via PCR. We have several recommendations that we could offer depending on your application. So for standard incorporation of barcodes and adapters via PCR, we would suggest our Ultramer oligos. These would be cater for your basic needs. And if you are using a long oligo like this, we would also always recommend incorporating 1 to 2 phosphorothioate bonds between the last 2 to 3 bases, and this is actually protecting the oligo from very active 3' exonuclease activity of the NGS polymerases. So just be careful when you're ordering the longer oligos. Then we also have a product which we can offer, which is HPLC purified oligos. This would be a bigger investment to make, but will allow you more surety if you can absolutely not tolerate any truncated products. And then I spoke about true grade processing if you are looking for clinical diagnostics and samples that are present, or variants that are present at very low percentages. Okay, and let's say you want to incorporate your adapters via ligation. We can offer you Y-shaped adapters according to the sequence of your choice. Normally, when you want a duplex Y-shaped adapter, you would need to include also one phosphorothioate bond between the last two bases, and also a 5' phosphate added to the indexed adapter to facilitate ligation. People normally ask me, what is the purification method that you would recommend for NGS oligos? And we would always recommend HPLC. And the reason for that is if you compare this to PAGE. PAGE is a process where you're excising a band from a gel, and it might allow some crosstalk to occur when you're doing a high level of multiplexing. So we always recommend HPLC purified oligos. For these adapters, you can have them delivered duplexed already, or you can have them non-duplex, so you can duplex them yourselves. True grade is normally supplied non-duplexed, and you can have an option of either dry down or resuspended. When you are thawing these, obviously, when they are duplexed, it's only about 12 nucleotides that would overlap. So when you go through multiple freeze-thaw cycles, you might have the strands dissociate. So you just need to be careful with handling these adapters as well. Now, just to talk about library prep efficiency, we've seen that the quality of your oligo does affect the library prep efficiency. So that's one of the reasons also why we would recommend for duplexes HPLC purified DNA. Here we achieve the 30% library prep efficiency. When you use Ultramers, it goes down to 70%, and for standard desalt, it will be even lower. So just do not compromise on quality when you are using and buying adapters for duplexes. So I've spoken about true grade processing, and here you can see a paper that was published by the Sanger Institute where they're looking at a high level of multiplexing, 384 different Illumina barcodes, and they were looking at very low variant detection, so variants that are present at lower than 1%, and they were looking to reduce the index crosstalk as much as possible. So they compared these adapters where they had HPLC or PAGE purified oligos, and the results that I obtained were 0.56 and 0.34% crosstalk. So when you're looking at variants that are lower than 1%, even losing that percentage of reads, it is still quite a lot. So they were looking to reduce that, and they ordered true grade processing from IDT, and they were able to dramatically reduce the crosstalk by 0.03%. And that's a 10-fold reduction. So if you're interested in clinical diagnostics and adapters for calling rare mutations, please ask us about true grade. So the solutions we would have for target enrichment, and as I mentioned, the building blocks for all our panels would be our xGen Lockdown probe. So these are 120 mer DNA oligos with a 5' biotinylation, and these have been synthesized with IDT's proprietary Ultramer synthesis pipeline. This ensures a high percentage of full-length product. And here, this explains the Ultramer synthesis pipeline. So if you are plotting during oligosynthesis, the percentage full-length product as opposed to the oligonucleotide length. I don't know how familiar you are with oligosynthesis, but normally you start with a starting yield, so the first base is already incorporated, and then you will add subsequent bases. And obviously the number of bases, if you have a starting scale, you would like 100% to be incorporated. Unfortunately, chemistry is not 100%, as you know. And the higher the coupling efficiency, the higher the number of oligos that will actually or bases that we incorporate in different cycles. So here you can see for a 99.5% coupling efficiency for Ultramer oligos compared to the industry standard, which is a 1% decrease. For 120 ml oligo, you would have 15% full-length product as opposed to 55%. So just decreasing the coupling efficiency by 1%, you lose 40% full-length product. IDT will also QC every single oligo with an ESI-MS trace, and we are a company that have invested in these ESI-MS instruments and allows us to QC all oligos up to 200 mers. So what sets us apart from the competition is the way we synthesize, as I just explained to you. So each probe will be individually synthesized, individually QC'd and normalized to the same concentration, and this will allow you a high uniformity of coverage. So for our exome panel, we synthesized 430,000 probes individually QC'd, generated an ESI-MS trace, and normalized at the same concentration. So we can show you these traces to be able to guarantee that the probes are included in your pool. When you're looking at the competition, an array-based synthesis supplier would synthesize all the oligos simultaneously on a glass slide. So if you're synthesizing that many oligos simultaneously, once the synthesis is complete, it has to be cleaved off the slide. So once it's cleaved, it's all pulled together, so you cannot individually QC. So why is this important? This is showing you the 7 to 10% random failure rate that you would get, irrespective of your platform or supplier, when you're synthesizing oligos. So when we synthesized our exome, we had to go through 6 rounds of synthesis to be able to include every single probe in the final pool. So for 430,000 probes, after the first round, we picked up about 31,000 failures. We QC'd those, we were able to resynthesize. Then we had about 2,100. We QC'd, resynthesized, QC'd again, resynthesized, and went through 6 cycles to make sure that all the probes are included in the final pool. For the competition, if you synthesize on a glass slide, on an array, you have random failures like this as well, but there's no way of picking this up, and there's going to be batch-to-batch variations. Essentially, IDT does not have any batch-to-batch variation because we QC every single oligo. So that is a very nice advantage that we can offer. We also don't have any issues with synthesizing probes with a high GC content or QCing. You can see a ESI-MS trace of the probe containing 78% GC. So what happens in the tube during in-solution hybridization? And this is showing a picture of one fragment containing the adapters and the blocking oligos hybridized to that. So our blocking oligos, as I mentioned, are universal. So in that sense, you are able to only use, irrespective of the number of indexes, one forward and one reverse blocker. Also, COT1, I'm sure you're familiar with that, but basically that is used to block repeat elements within the target regions. So your oligos, lines, and sign elements. If you exclude COT1 and the blocking oligos, we have seen that you can reduce your on-target percentages by 10 to 30%. So it's really essential that you would include this. Then you would have your lockdown probe with a biotin, and then you have obviously your pull-down method here, strip and magnet, and the magnet obviously. So how do we design? When we receive your target sequence, we would put that through our online design tool. And the first step would be to do a repeat masking. So we do not design probes across repeats. We also then do a 1x end-to-end tiling, which means that every target base is covered by at least by only one probe. And the first probe is sitting next to the second one. So there's no space between, no overlap. So yes, you heard right. We are confident enough to tell you that the 1x end-to-end tiling design will allow you a high uniformity of coverage. And that is why you will have, it's exactly the way because we synthesize, the way we synthesize our probes. For some of the competitors, they can go up to a 30x tiling. And why would they do that? Exactly what I referred to is the way they synthesize. They cannot QC, so they're covering themselves to make sure that the probes are actually in there, and overloading the panel with probes. So as I mentioned, the lockdown probes would form the basis of our panels, and here I would like you to break down the solutions we have into custom and pre-designed. So pre-designed would be for the human species, and custom would be for all other species than human, and also for regions that fall outside the pre-design. So any custom panel we can design for you. Then for pre-design we have gene capture pools, so almost all human genes are represented. We have pre-designed panels of probes available that would target these genes. And then you can also select from validated stocked panels, which will include an exome panel, an AML panel, pan-cancer, or an inherited disease panel, which is essentially the Mendelium. So let's talk about the cost efficiency and the formats for the custom solution, so all other species than human. Let's say you're interested in a panel, custom panel. You have three sizes to choose from, either minimum, mini, standard, or XL. And depending on the size, you have a minimum order attached to that. So for a mini, you would have to order 200 probes, and you would be delivered 3 picomoles per probe. So just to give you an idea of what the target space would be or size would be for 200 probes, you will multiply that by the length of a probe, which is 120 mers. So you will have about 24 kbs. Now, when you order the mini panel, you would receive 200 probes at minimum order and receive 3 picomoles of each probe. So you'll have 600 picomoles pooled into one tube. When you do a capture, you would only use 3 picomoles of the entire pool. So this will allow you for 200 probes to do 200 captures at 10 euros per capture. So this is really cost effective. When you order 1,000 probes, you would be able to do 1,000 captures, and that's where one reaction per probe comes from. This is not even taking into account multiplexing. So if you're multiplexing 10 samples together per capture, you will pay €1 per sample per capture. This is just showing our online design tool, and this is the way you would go about designing your panel. It's a very easy process. If you're using the human, mouse, or rat, you can use the online design tool. You would have for specific bed, you would have specific input formats. You would have a bed, file, gene symbol, or ref-seq numbers that you could put in. Even faster sequence if it's other species. So you can go through different steps. You can add and select our lockdown reagents, which would be our wash and hybridization buffers. Here we have either 16 or 96 capture sizes available. You can also choose your blocking oligos. Here we just need to know which would be your adapters and platform that you're using, and you can select these pre-designed blockers based on the adapters. You can also have a custom option. So if you have customized your adapters, we can also design custom blockers for you. The blockers are very beneficial in that they can improve on-target percentages per run quite significantly. So here is a Foundation Medicine comparison where they looked at standard blockers compared to xGen universal blocking oligos, and they found that in some runs they were able to improve on-target percentages by up to 60%. So really, if you're improving on-target percentages, you are actually saving money because you're generating more usable sequence data. Okay, so that was the custom solution and the design tool. Now the pre-designed solutions we have, first of all, would be our gene capture pool. So if you're interested in human genes and the coding regions of human genes, this would be the solution to go for. Here you have the option of having your genes pooled together in one tube, or let's say interested in 96 genes, or even more on different plates. You'd be able to have 96 different gene pools targeting one gene per well. So you can mix and match and put together your genes as you go along and as your sequencing needs change in the lab. So as I mentioned, these are targeting coding regions, and you will pay a flat price per gene irrespective. So you'll have pay a flat price per gene irrespective of the size. So even a small housekeeping gene and a dystrophin, which is a very large gene, would be the same price, and the turnaround time is very quick. Just to show you some functional data, so this is a small panel, and if you can remember, I spoke about what to expect with on-target percentages when you're looking at specific target enrichment of small panels. This is a small panel of 71 kb, only 20 genes, 805 probes. So what we did is we did an enrichment on 6 samples and sequences on a MySeq with 2 times 300 reads. So the on-target performance that we obtained, now normally you would expect between 25 and 35% for a panel of that size, but we were able to achieve, excluding the flanks, on-target percentages of about 50%. When you include the flanks, it is just above 60%. So really nice result. When you look at the read depth, we were able to have more than 95% of our bases covered at about 200x, so really good result. So that just gives you an idea of what to expect with our gene capture pools. Then we have pre-designed panels available, and here you have the 4 panels that you can choose from. You can see that they differ in content and target size. And the exome panel basically is a 39 megabase in size, so it's a small non-inflated exome targeting about 19,400 genes. So the result that we obtained, we were able to show that the lockdown probes cover the exome completely. So this is an exome that we compared to 2 versions of an array-based exome, and obviously what you want here is as high a coverage depth as possible at 100%. So the higher the coverage depth, obviously the more percentage you want covered. And you can see that how the IDT performs better than the array-based competitor. Here you see the same comparison, but it's showing the exome uniformity. So here you want a nice bell-shaped curve. You want a high percentage of your target bases covered between 50 and 150%. And here you can see what we achieved with IDT compared to 2 array-based exomes. And obviously you don't want any coverage at the extremities, so you can see the IDT is almost non-existent at the extremities compared to the array-based system. The fact that the exome has a high uniformity of coverage and also a small exome allows you to multiplex more exomes together. So this was multiplexing 31 exomes together on one lane on a High-seq 2500, and compare that to a smaller array-based 37 meg exome. Because the uniformity of coverage is not as good as IDT, you were basically able to multiplex less within one run. So you need essentially much more sequencing for the array-based systems compared to the IDT system. So some advantages of our system, I spoke about uniformity at length. So the way we synthesize our probes allow you to achieve a high uniformity of coverage. And here is a comparison that Foundation Medicine performed, comparing the IDT probes to an RNA-based bait system. So for the Foundation 1 panel, they need to be able to confidently call mutations more than 99% coverage of target space at 750x. And here you can see for the IDT panel, they were able to achieve that. But for the RNA panel, it was sitting at about 300x. So you essentially need double the amount of sequence for the array-based system to be able to achieve that depth of coverage compared to IDT. We are also very flexible, so depending on your needs, you could start building up your panel in your lab and add content as you go. So let's say you're adding, you have a custom panel and you want to add some gene capture pools, you are free to do that. You can also spike in our panels into competitor panels. So it's a very flexible system. We know exactly what concentration of probes would be in the pool, and we can help you and guide you through the steps to take to be able to mix and match and spike in these these different solutions. And here is an example of what we did in-house. So we had the pan-cancer panel, which we then sequenced, and that was the coverage that we obtained. Now we wanted to show that spiking in probes with different content would not influence the result. So here you can see we spiked in 103 probes covering 3 genes. We spiked it into the pan-cancer panel, and you can see that had no dramatic effect on the coverage of the pan-cancer, and you can also see complete coverage of all the regions of the spiked in probe. So this was a really good result, and competitors would take 6 to 8 weeks to accomplish this because they have to synthesize an entirely new panel. Turnaround time is very quick, so you'll receive your pre-designed panels and probes within 2 weeks. You're able to test it and then order supplementary probes, so you can spike into your existing panel. For competitors, we've heard anything from 6 to 8 weeks. You then test your panel after that and then have to order an entirely new panel that's optimized because they have to do a synthesis from the start. The protocol is very quick, so you can complete the protocol in 9 hours, and this is the capture step. We use a 4-hour hybridization at 65 degrees that have shown to significantly increase the on-target percentages. Now to finish off with some applications, in the literature it was described that when you are looking at very rare mutations, it's very important to make sure that you have the coverage that you need. So when you are looking at variants that are present below 5%, you need about a coverage of 500x. And keeping that in mind, this was a study that was performed where the researchers were using a competitor exome, the NGS exome, and they were looking at AML content. Now AML mutations are present at very low percentages, and you would need, as I just mentioned, about 500x coverage. Now when using the NGS exome, they were able to achieve only about 140x mean depth of coverage, which is not good enough to call AML mutations correctly. So what did they do? They spiked in our AML panel into the Roche NGS exome, and they were able to increase the mean depth of coverage in excess of 500 and also the coverage to 100% for 20 and close to 100 for 40x. So this immediately put them into the ballpark and to the region where you can confidently call the AML mutations. So this is a nice example of a IDT panel being spiked into a competitor panel to be able to rescue those regions that you have dropouts and improve the coverage. You can see another example where the researchers were studying the third promoter, which is a promoter that's implicated in disease, melanoma and bladder cancers. You can see for whole genome sequencing the coverage obtained. An array-based system, the coverage, and compared to the IDT, you have a much higher mean coverage and fold coverage when compared to those two different applications. This is another example of improved coverage using IDT when you're looking at translocation. So this is an old intron 19, which is a very difficult intron to study. You can see the coverage obtained with RNA probes compared to a complete uniform coverage when comparing. And you can see it almost looks like Table Mountain, which reminds me of home. Also, Capture-Seq is an application that is very quickly gaining traction. For being able to pick up specific gene fusions, you need a system that is able to detect the expression levels at different dynamic ranges. So here you see 3 orders of magnitude where these different fusions are expressed, and the results obtained, so IDT's capture panels were able to detect all the different gene fusions at different expression levels. Copy number variation is also possible if you have high uniformity of coverage. So let's say you're looking at the target region where you have 500x coverage for your entire panel and suddenly see a drop of about half. It does point to the presence of a deletion. And we also have customers that have reported the possibility to detect single exon deletions. So really good to hear that as well. Something that I want to touch on is duplex sequencing, and this is a paper that was recently published. Here the researchers were looking at specific ABL mutations that are very rare. So when sequencing Any mutants that are present below 1% is very difficult to call because of the noise. So what did they do? They ordered these Y-shaped adapters from us with a randomized duplex tag. These tags would mark individual ligation events. So you'll be able to eliminate PCR and sequencing errors and DNA damage. And to be able to remove all those duplicates, you will significantly reduce the noise, and you can see the results. So they're able to pick up a mutation or rare variant that's present down to 1 in 10 to the power 7. So very good result here. We also verified this in-house. Here we have a poster that was recently presented at ESHG in Barcelona. We were looking at the low frequency variant detection from FFPE and cell-free DNA using our target capture and also our unique molecular identifiers. And what we found is that our capture technology is compatible with DNA of varying quality and quantity. We were able to uniformly enrich DNA targets to facilitate detection of mutations between 1 and 5% with a 500x million target coverage. And you can see here the percent mean coverage, which is reproducible for specific sample types, cell-free DNA and genomic DNA, high quality and low quality FFPE. So if FFPE and cell-free are very difficult samples to work with, degraded, low input, we were able to start off with as little as 5 to 10 nanograms of cell-free DNA and as little as 25 nanograms of FFPE samples. And we also included UMI, so we were looking to see what the effect the UMIs would have on the calling of specific mutations. You can see the background once again, and in red and green it will be the specific true variants. And this is before UMIs, this is after inclusion of UMIs. So dramatic reduction of noise, and we were able to detect variants below 0.5%. So I just want to finish off then with some customer testimonial, and this I'm sure you're familiar with Sophia Genetics. So it's a company that's making a lot of noise in the genomics market. They're the leader in data-driven medicine with their bioinformatics software. They were evaluating several target capture technologies to be able to include that as a solution. After going through that evaluation period, they decided that the IDT probes were the way to go. So with our xGen technology, we've collaborated with the analytics platform of Sophia, and we are able, and this is a slide that I've taken from Sophia Genetics talk that they gave at ESHD. They were able with their platform and our target capture to produce 100%, and that's their words, 100% sensitivity, specificity, reproducibility, and repeatability. So really nice customer testimonial. If you are interested in the validated solutions that Sophia can offer, please look at their website, and if you need their contact details, just let us know as well. So I hope that I was able to bring across the message on how we synthesize our probes and what sets us apart from the competition. So we synthesize obviously individually, we QC and normalize individually, and this will allow you the high uniformity of coverage that you would need. Our blocking oligos improve the on-target percentages significantly. We have several solutions available for both pre-designed and custom. We have a lot of flexibility. We have a fast turnaround time and protocol, and we have shown success in a lot of applications. And with that, I thank you very much for your time.